So I have a question for Dr. Louter. I was listening to you, and something that strikes me is seems to be a conflict of interest between, or potential conflict of interest between the people that is giving you the information and how much that information they are giving you, how much you can trust or not, because it's their own interest that is going to be at risk. So I was wondering how you are handling that part of the problem.
Conflict of interest in terms of, well, our in terms of protecting confidentiality.
Well, I was two parts. One is the group or the whatever investigators that they have a personal interest on making sure that this product moves forward. So they have a conflict of interest in a sense of giving you the right information. That's the main thing I was wondering.
Well, okay, maybe I should have. Okay, there's one. I should have emphasized one thing also, and that is a part of the regulations that I deal with has a research and development exemption. So right now in Canada, anything that goes on at with large animals, nothing needs to be reported to the government as long as it is fully contained. So if the animal doesn't get out and its genetic material doesn't get out, and nothing associated with toxicity of the organism doesn't get out, then there is no need. It's completely exempted right now. We are actually, though, re-looking at that particular aspect of our regulations and have been talking to scientists and researchers in Canada about a reporting requirement that we wanted to add to our regulations. But currently, it doesn't matter who is working, you could do anything you want, in essence, in Canada now in research. If it doesn't get out, then there's no need to report. But the other side of your question dealing with confidential information. When we publish a risk assessment summary on our website, it's done only in consultation with the proponent and only after all the confidential information has been removed from it. So it's an effort with a proponent to be more public about the risk assessment decisions we've made. But at all times, any confidential information, I would say that's true for all the regulators here. Anytime information is given to a government entity, it's given with the premise that it will be kept confidential. That's the basis by which you operate as part of a government entity.
Hi, Alison Van Inen from UC Davis. I think this is perhaps the question that's most up on my mind, and I'll ask it to all of you because I'm not sure I have an answer, or I didn't understand the answer to this question. If I make a single base pair change using genome editing, is it or is it not regulated by your agency? And I guess I'll just go down the line there and put that question to each of you.
USDA does not regulate it.
The FDA is currently deliberating on that, so we do not have a response for you right this moment, but I know we are actively working on a response to that question.
It could be. If it's part of a research program, though, and it's fully contained, it wouldn't be. So if you wanted to release it or commercialize it, then it would be a different story. Then we'd start at asking you for more information and have something we call a little consultation to figure that out.
Well, in the case of Argentina, if this modification was done on an animal of agricultural interest, nowadays it will be regulated. But if it is done in a crop, you have these new regulations, the 133, and you, as an applicant, can request Canaria to determine whether it falls under GMO's rules and regulations. So maybe no, probably no. In the case of the crop, maybe in the future it will be the same criteria will be applied for animals.
Steve Nemi, Harvard University. I'm here, though, representing the American College of Laboratory Animal Medicine as their ILAR roundtable representative. I have two regulatory clarifications I'd like to pursue. First, to Dr. Carpenter. Dr. Harper, it's my understanding that no USDA APHIS policy has regulatory standing or can be enforced. And I've asked that question numerous times before, and the answer has always been that they are not enforceable. Is that your understanding as well?
Well, actually, I can't speak for APHIS because I work for Agricultural Research Service.
Okay.
But no, the policies are guidance to help you to determine what you need to do to meet the regulatory standards.
Sure, thank you.
Mhm.
Okay, and then for Dr. Wileman, I'm still wrestling with this scenario. If I develop a pig model of human colon carcinoma and is to be used for drug screening, it will never be a commercial product. It has no therapeutic or diagnostic value per se. But in your scenario, that was an animal that was genetically edited that still had to be reviewed by FDA, correct?
Sorry, so if it was, if the editing inserted an rDNA construct, at this time it would fit our current paradigm for regulation of that animal.
And what's the rationale for an academic center, for example, having to file with USDA for an animal that is purely used in a discovery mode? Excuse me, FDA.
Right, so we would still, under the INAD requirements outlined in 21 CFR 511, we would still like to be informed about the existence of those animals and any shipment of animal or animal materials that's happening, even to other collaborators that you may have for further studies on the animal, to be able to address your specific requirement or the studies on those animals. At this time, we would still require you to open an INAD application with us.
Okay, and does that apply to mice as well?
No, we do not regulate rodents, mice, or rats
Okay.
because at this time, as it was presented with the current regulations that are in place with the NIH Biotech Office, for instance, they have sufficient oversight of research going on in rodents, so FDA is not involved in that.
Okay, thank you.
This is Susan Silk again, but once again, I'm not speaking as myself or for OLA. I'm reading questions from our colleagues on the web. This appears to me to be directed at APHIS, so I don't think we have the right expert on the panel to answer it. With the use of genetically engineered animals expected to increase in the U.S. as a result of gene editing tools described, is the USDA considering adding to its annual report a column that indicates genetic status of animals? And they put in parentheses, not genetically altered versus genetically altered, to better understand the impact of genetic modification and to quantify the number of genetically modified animals. This is carried out in other countries and would be important to collect to improve transparency about this research.
And I don't think any of us can answer that question because nobody represents APHIS today, unfortunately.
Hi, my name is Tim Schwab from Food and Water Watch. And I wanted to revisit a question that was already asked about conflicts of interest. And I think you partially addressed it from the Canadian perspective. I wouldn't mind if you expounded on that, and also if the FDA addressed this. But when FDA regulates a genetically engineered animal, all of the data it reviews come from the sponsor company, which has a clear financial interest in presenting its product in the best possible light. And there's been several instances where we found out that FDA didn't have all the information it needed. So, for example, Aqua Bounty salmon had a major disease outbreak in 2008 or 2009. FDA apparently didn't find out about it until 2011. So this kind of, and it seems like the way that the regulatory paradigm is established to trust the sponsor to provide all the data, it just kind of creates a scenario where you're going to miss some things. So can you address that, please?
I don't think I'm in a position to speak directly to the Aqua Bounty salmon. I know there's a presentation tomorrow and there's a lot of information on that product Since it's approved, it's on our website, and our press office will be able to provide further specifics on that approved product. I would say that we do have mechanisms in place in our review of data when we are doing our evaluation of not just the submitted data and information towards an application, but information from the field, information in published literature as well, and inspections that the FDA does. At which time we also review, as I indicated in one of my slides, I think talking about phenotype, biosurveillance, all of that, those aspects. We look at veterinary treatment records, daily health observation records. So if there was something that happens at any facility that has GE animals, that is something that FDA would find out even during its inspections. And inspections are carried out before the approval of a product.
But just to push back a little bit, on a pre-commercial level, there's not likely to be much independent scientific literature that you could consult. So just given the reality that you depend so heavily on sponsored data to make a very, in this case, precedential scientific determination, how do you really have the confidence that you really have all the data you need?
Again, I will stress back to the inspections. We also do, during the time of the evaluation of the phenotypic evaluation for the GE animal, we do conduct an inspection even at that time. So there are multiple opportunities that the FDA has to go at the sponsor facilities where the animals are raised or maintained and have the opportunity to look at lab notebooks, unedited data, etc. So there are avenues in which FDA is able to pursue that.
Hi, Carolyn Plunkett from the NYU Division of Medical Ethics. I'm interested in particular in insects, genetically edited insects that are regulated under the FDA regulations as opposed to APHIS. And as part of the gathering the safety and efficacy data, it seems that caged and limited releases would be required as part of the research process. So I wonder, not just sort of the approval for ultimate marketing and licensing, but what kinds of guidelines or rationale is there for approving releases of, again, in either caged or like an open limited release exist?
So again, I'm not able to address specifics on a particular application or a particular product. What I gave was a very general overview of the kinds of risk questions we address in evaluating various aspects of the application for the particular product. With respect to insects, again, I know that there is some information on our website, and we can always urge you to follow up if it's something you are pursuing to discuss with us subsequently after the meeting as well.
So I hate to keep picking on the FDA, although I don't think this is picking. I've got 3 questions, but I think they're each pretty short. I just want to confirm that I understand it correctly. First, FDA, as I understand it, has no authority with respect to non-animals. Is that correct? So plants, microbes, et cetera.
No, we would not be doing, well, see, I represent the Center for Veterinary Medicine, so I know at our center it's animals that we're working with.
Yeah. Well, your statute talks about the structure and function of the body of man or any other animal. So
so there are aspects of GE plant regulation that fall under Center for Food Safety and Applied Nutrition at the FDA, and there are divisions within USDA APHIS, I think, that would cover that as well. It would not come to the Center for Veterinary
with respect to, so there may be some food regulation, but in terms of drugs, devices, biological products, non-animals probably are not covered by FDA?
So I can probably add to that question. So Dr. Diane mentioned in our discussions that a moth, genetically engineered moth, which is considered a pest, will be regulated by APHIS. Yes. An Oxitec genetically engineered mosquito, which can affect the health of humans, will be regulated by FDA. So that can be a drug.
Both of those are animals, but okay.
So, but there are overlapping. I showed you the diagram, so there are overlapping.
So second question, to the extent there are do-it-yourself biohackers who are not introducing their products into interstate commerce, does the FDA have a position on whether they're covered?
I'm not the correct person to address that. we have an Office of Compliance that gets involved as appropriate.
Okay, and last question, I promise. And it's really the glowfish regulatory discretion that made me wonder about this. Is there a policy with respect to pets or animals as genetically modified art that the FDA has decided upon to follow?
Genetically modified art?
You wouldn't believe it. Yes. There are geno artists who are very interested in doing a variety of strange things. I think the most famous, although he really didn't do it himself, was the GFP bunny that had GFP, green fluorescent protein, moved into it. That was produced in France, so it wouldn't fall under the FDA. And for better or for worse, it turned out it didn't get into the fur. So the only things that glowed were the tip of the nose and the pads of the feet and the whites, the greens of the eyes. But if somebody were to, say, dog breed, and were to put in a substantial chunk of recombinant DNA, so avoiding the issues about whether it's just a deletion or a substitution, has the FDA thought about, to your knowledge, whether or not it would view that as subject to its regulation?
So again, we would be involved in, for instance, as you said, there is actively an rDNA construct used in the engineering event for that particular dog. So yes, that would be a regulatory trigger for the FDA, but I would be curious to learn more about the art aspect of it, and maybe we can talk offline.
Hi, my name is Evan Bromfield. I'm at the Center for Food Safety. So say I'm a young guy, been dating my girlfriend for about 3 years now. I'm thinking about proposing. I get down on one knee and I have 3 rings to give to her. They're all 3 diamond rings. One was 3D printed in a box in a factory. One is a blood diamond and another is a certified fair trade diamond. I think in this instance, everybody's going to agree that my girlfriend would choose the certified diamond. It's a question, in my mind, that's a question of process versus product. So if that's true when we're talking about a diamond ring or something like that, can you please defend that same position when it comes to biotechnology?
I assume you're directing that to the FDA?
I would like to hear from everybody.
I was curious. I mean, so so for one thing, is the size of the diamond same on all
Yes.
those 3 rings?
They are, yes.
Because perhaps that would be important to your girlfriend as well.
It's the exact same diamond in all 3 cases.
Not a drug product for us, not a regulatory trigger for me to be able to address that.
All candor, I think the question is, the position that FDA took is we're going to regulate the product, not the process. So in this question, you're asking is sometimes the process may provide exemptions, or maybe
Sometimes the process matters, and I think everybody's kind of said that in their regulatory opinions. We're not regulating based on the process, we're regulating based on the product. And I just want to hear you kind of defend that philosophy, as it were.
Well, in all seriousness, I think the best way to address that for me at this time is you might be aware that there are discussions going on even for the coordinated framework and the discussions of emerging technologies in that context as well. We've already had one meeting. I think it was on the 31st of October. I believe there's another one scheduled in a few months. And I urge you to continue to stay tuned into that. And those are public meetings as well. So that would probably be the best forum for you to have these discussions.
Right, as the moderator, I get to jump in. And I would actually like to address that question. And I think that the process is regulation of the process is already in place. As the USDA representative pointed out, animal research is regulated the process before you even start, how that animal is made and what it's going to be used for. The process, whether it be gene editing or anything else, is really irrelevant to that approval process. So I would argue that that process is already in place of oversight of the process itself.
So Scott Farnkrug, Recombinetics. I did notice there was actually a theme that was maintained by all of the regulatory agencies that kept pointing to whether or not something is new or novel. So in Canada, If it's not a novel food, then apparently it doesn't fall into the regulatory paradigm you're operating under. We have something called the investigational new animal drug mechanism at the FDA, and I noticed even in Yanina in your presentation, the focus was on new or novel food, for example, and so I wonder, people have been dancing around it here, even a story of rings. If something has a genotype that in fact is not new or novel as a product, regardless of whether or not we create those under procedures that are consistent with animal welfare, of course. Is that going to be at least a handle for us to make decisions, for you to make decisions on how you manage or regulate products that are presented to you? Is novelty really the issue? And if it is, then boy, I'm feeling a lot better about my stock. Livestock, that is.
So what was your question? Was it like what's novel or what's
Right, seems to be a predicate for all of these regulatory paradigms, that whatever we're talking about, has to be different, has to be novel, has to be new in order to justify the regulatory consideration and review. And I guess we just have some very clear examples here where we're replicating genetics that already exists, and so they're by definition not novel.
Well, you'd still have to show that, I think. So I think the more theoretical, I'm going to say theoretical question because I don't want to pronounce on that, is that if there's a modification or a technique used, but the result has no genetic footprints, then and is completely utterly indistinguishable, then I think you've probably got to that point where you could say that that's not a novel product at that point. But you might have to prove it.
Easy.
Again, in Argentina, at present, you will be under regulation. Your product will be under regulation, but maybe in the near future we'll discuss within the agencies on the criteria that will be applied for this kind of product. So yes, maybe it will be covered by the regulation, but now I can tell you.
For FDA, I just want to clarify the new animal drug provisions in the Act and the Code of Federal Regulations. They precede anything that has to do with GE products. So the new in our new animal drug provisions comes from the Act and the regulation. And what we've tried to do for the GE animals is try to clarify how that applies to the products, but we are not necessarily calling them new products, and that's not where the new in the new animal drug provisions has come from.
But in fact, it is new animal drugs. It's a new drug that you're regulating, right? You're not gonna redo Tylenol.
So as I said, it's really the new. Okay, I think our legal counsel wants to address that because she.
Sorry, Malini, thanks. I just thought I'd jump in since I'm a colleague of Malini's from FDA. New under the Federal Food, Drug, and Cosmetic Act does not mean what you ordinarily would think of as new. It has a very specific meaning under the Act, so it applies both for animal drugs and human drugs. Human drugs, it's a new drug, and for animal drugs, new animal drug. I don't have the entire definition in front of me, and it's quite lengthy, but in a nutshell, it's a drug that's not new has to be generally recognized as safe and effective and used for, now I'm forgetting the language, significant time. And that also has a very specific meaning in that you have to have data that's been public. And so really, you'd be hard-pressed to find an animal drug out there that's not new, and the same is true of human drugs. So when you ask about a human drug that's been out for decades, it's most likely still considered to be a new drug.
So there's no such, then why use the word new?
Congress wrote it, we didn't, so.
It defies logic. So.
Yeah, hi, Dave Anderson from the University of Washington. I have another question from the virtual community. Taking some liberty to paraphrase this, it says, is it correct to conclude from Dr. Louter's presentation that Canada approved the Aqua Bounty salmon in 120 days, whereas the US FDA took many years?
It was 120 days from when the information was deemed complete. It still took a while to get to that point. So there can be a lot of going back and forth with a proponent till you get to that point. But once you've hit that, my dossier is full button and that green light goes on, then that clock starts. It's part of our legislation. So I really can't, it's not a choice that I was involved in making that. That's just the regime under which I operate.
Okay. Thank you.
Okay, Alison Van Eenenham again. I guess I'm still struggling a little bit with Scott's questions here and this whole concept of a drug. And when it's a recombinant DNA construct making a protein, I get that. If you've got a single base pair change that replicates an existing mutation, the myostatin, for example, what exactly is the drug and in what way is it new if I could have achieved exactly the same genotype and phenotype by introgression from, I don't know, Belgian blue or whatever? That to me is, if there's no rDNA construct, then what exactly is the drug?
My apologies, but I really cannot address that definition at this point for that particular type of change. As I said, since the agency is under deliberation on that particular question right now, I am just not in a position to comment on that. But stay tuned, and also, as I told somebody else in the audience, also continue to comment on and listen into the emerging technologies discussion as part of the coordinated framework discussion. And we are optimistic that we'd be able to provide a response to this in the next few months. I just do not have one right now.
Okay, are there any more questions? All right, I want to thank our speakers for this afternoon. You guys can go ahead and take a seat. So I'm going to give just a brief wrap up. I know it's been a long day, and everybody's ready to get out of here and enjoy the rest of their day. And so what I'm going to try to do is just give you a brief synopsis and interject maybe a little bit of my opinion. So you have to pardon me that I get a little bit of artistic license. I am a scientist. The talks this morning I thought were just invigorating. I mean, the potential with this newer technology is just amazing. And from a scientific standpoint, I am just excited about what the future holds. But I'm also a veterinarian, and my oath is to do no harm. And so I wear multiple hats in my job, and as the veterinarian, and especially being a laboratory animal veterinarian that's involved in regulatory oversight, I understand and I appreciate the role that the regulators have in this whole process. I am very thankful that where I am, we do not write policy. I would hate to have to write policy, because you've got everybody wanting a little piece of that pie. So what I want to try to do is just give a summary, hopefully a summary of what we did today. So we started off this morning talking about the science. And, again, this is amazing technology and what potential it holds. And there are a couple of comments that I think are very important to take home. And these two are the ones at the top, that is very simple. This process is nothing new. It's been going on in nature for a millennia. We are learning to do it more efficiently. And I loved Scott's comment on the fact that we're eating genetically modified plants and animals every single day, and we don't get sick. But what I think was presented forth is the power that this technology holds. That, with the zebrafish, the fact that we can get a much better understanding of early embryogenesis in this animal model, and then start, specifically targeting different genes. We get rapid and efficient gene targeting, which leads to less money, less time, less animals. And I did put a question mark, but I do believe that eventually it will, and more research. And so that allows the scientists to focus, perform experiments more efficiently, for less money, less time, and then that money can be used to further their research. Biofarming, actually producing these biologicals in large quantities that we have shown are beneficial in the treatment of human disease. We're finding that this technology is not only beneficial for people, but also beneficial for animals. And as we move forward, we need to remember that. This technology is going to allow us to make better animal models, more specific animal models. Especially, the mouse was great because we could go after that understanding of a single gene defect. But as you saw, trying to make these understanding the polygenic disorders is very difficult, especially from a breeding standpoint. We now have the ability to do this much more rapidly. And making these significant, these specific alterations. And one to think about is actually reversion to a wild type, is being able to correct a defect. Oops, uh-oh. Let's see, hopefully my battery will not run out. Let's see if there's a plug up here. Okay, we'll go on to the next one. All right, government. And I don't mean to simplify their role in this. Because, again, the scientist in me, I'm jumping and going, wow, this stuff is so amazing. Plus, the government speakers were in the afternoon, and I didn't have a lot of time to add to these. But they have a very arduous task in writing policy and oversight in these. I mean, first and foremost, they want to advance this science and the research. I mean, the federal government, the NIH is the largest funder of biomedical research in the world. There's nobody could argue that the government wants to get in the way of research. But they are the ones that are liable, in a sense. They are the ones that the general public comes to and says, you got to promise me that you're going to look out for me. And so they're the fall guys in all of this. They have to make sure that we protect the population. They have to make sure that they're protecting our agricultural commodities, both plants and animals. They have to make sure that it also will protect the environment. And I put in here animal welfare, not because scientists don't care about animal welfare. That is completely incorrect. I have never met a scientist who don't truly care about the animals that they're working with. But the government is responsible. Oops. Uh-oh, my battery just went out. Okay. All right. So the government is responsible for, making sure that the regulations are in place concerning animal welfare. So we're all on the same page and we know what to do. All right, the next slide is one that was briefly touched on some of the talks today, but that is the public, the general public. That is a group that needs to be part of this discussion because they're worried. They're concerned about what this means to their health, what it means to their environment, and what it means to the animals that we're using. Are we going to be creating or doing harm to these animals? Animal research is a very touchy subject to talk about. It is a very critical and important part of biomedical research. But if you ask the general public, would you support animal research if it resulted in pain or distress of the animal? Actually, the majority of Americans, at least, say no. And we have to be able to convince the public as to why we're doing this and the importance of doing it. And then finally, what I included in there was the animal. And some of the speakers today included that as well. And I'd listed up there basically the 5 freedoms. Freedom from thirst and hunger, freedom from discomfort, freedom from pain and distress. See if I can remember all of them. Freedom to express natural behavior, and I'm missing one more. anyway, it's important to include that in this discussion. And basically I had a Venn diagram. I brought those all together. And really, in my opinion, for this to move forward in an efficient and an effective manner, is you got to get where all of those come together. Otherwise, you're going to meet a stumbling block one way or another along the way. And then finally, my last slide was, and I forget the Greek mythology story, the man pushing the boulder up the hill. Sisyphus. And that boulder I'd put in there is public perception of genetically modified organisms. I do really think that that is one of the biggest hurdles to overcome. You know, the approval of the aqua salmon, and I hope to not take anything away from Dr. Stadish's talk tomorrow. You know, as a scientist, I perfectly understand what's going on with that and think that it is an excellent way to move forward with an agricultural product. But I was actually online reading some of the comments about the New York Times article that was published about it. And it really voiced a lot of the public perception, or really the uncertainty that they have with what's really going on. And I think that's our responsibility, is just to educate them. I think once we educate them, then they're going to realize that really, for the most part, what we're doing has been going on in nature for a long time. And with that, I'll get off my soapbox. I want to thank you all for coming today. Tomorrow, we will get into the ethical considerations. I think it will make for some very good discussions tomorrow. So we will see you all tomorrow at 8:30, I believe. I think so. So thank you very much.